In this video, we're going to look at how to solve a Punnett square with two sets of genes, which basically means we have two alleles. And this is a little bit more complex than the previous one we looked at. And let's take a look at how we do this. We're going to first describe the parents. It says parent 1 is heterozygous for a codominant A gene. And we're going to represent that by a capital A. And it says they're heterozygous for Rh positive, which means parent 1 is going to be AO plus minus. Now this is blood typing, and this is probably the most complex one we can actually get with it. It says parent 2 is heterozygous for the codominant B gene, which means they are going to have a B gene and they are going to have an O gene, and they're heterozygous for Rh positive, which means that they have got a BO plus minus as well. Now, with each parent, we've got to look at what the possible gametes are. So when we look at this, we're going to have in each set a copy of one of the A alleles, or the O alleles, or the B alleles, and a copy of a plus or a minus. So it says that we're going to take an A from over here, and we're going to take a plus with it, and that's going to give us an A plus on one side, which means the other side has got to be an O minus. The other possibility we could have is we could have the A pairing with the minus, which would give us the O pairing with the plus. When we do this with parent 2, we get the B pairing with the plus, which yields the O pairing with the minus, or the B pairing with the minus, which would leave the O pairing with the plus. Now that is what we've got as possible gametes for each one of these. Now we're going to take this over to the Punnett square. In the Punnett square, it says across the top, we've got the possibility of an A plus, an A minus, an O plus, an O minus. Down the side, we've got the B plus, the B minus, the O plus, the O minus. Now, when we add these together, we're going to be careful because we want to put the A before the B, and we want to put the plus before the minus. Doesn't matter which side they come out with. We also have the A before the O and the B before the O as well. So it says over here we're going to take the A and the B and the plus and the plus. Over here it says we're going to take the A and the O and the plus and the minus. Now notice, it doesn't matter which one has the plus, we're going to take it from that particular one. And then we start filling in the chart with all of the other possibilities in here. And as we fill in the other possibilities, we get a total of 16 different ones. Now we go from there where we've got all of our possibles, and we start looking at genotype and phenotype. Genotype, we said, was the actual genetic makeup. So we take our Punnett square here, and we are going to look at the possible genotypes, and these are all of the possible genotypes that you've got out of here, and then we've got to figure out how many of each one we get. It says the AB++, we've got one of them. The AB plus minus, well, we've actually got two of those. The AB minus minus, it says we've got one of those. The AO++, we're going to have one. The AO plus minus, it says we're going to have two of those. The AO minus minus, we get one. The BO plus plus, we've got one. The BO plus minus gives us two. The BO minus minus, we've got one. The OO plus plus, we've got one. The OO plus minus, we've got two. And the OO minus minus, it says we're going to have one. And that's the possible genotypes. When we start looking at the phenotypes, remember, A is dominant, A, B are codominant, plus is dominant to minus. So in here, we're going to start looking at the possible phenotypes as being AB positive, AB negative, A positive, A negative, B positive, B negative, O positive, O negative. So we start adding these together. The AB, if it's got 1 plus, is going to be AB positive, so we've got 3 of those. The AB negative, we've only got 1. The A positive could have an AA or an AO, and it's going to have 1 plus, so in this particular case, we've got 3 of those. The A negative is going to have an A and 2 negatives. The B plus is going to have a B and a plus, and it says over here we've got 3 of those. The B negative is going to have a B and 2 negatives, and we've got 1 of those. And the O positive is going to have 2 O's and 1 positive, and it says we've got 3 of those. And the O negative, it says we've got 1 of those. So when we look at this particular makeup, you can see that we've got a lot of different genotypes, we've got a lot of different phenotypes. But in here, our phenotypic relationship is going to be 3-to-1 in each one of these little groupings. There are 4 groupings, which give us 16. There were 16 different genotypes, so we check our math in there to make sure that we've got 16. So when we look at these, we can see that there's a tremendous variation, but what we get is we put this together in a rather simple method, and we can find out what the possible reactions could be with each one of the different sets of gametes.